Good morning, everyone. Today I am here to read a chapter Enzymes and Co-enzymes. So let's see the introduction. Enzymes and coenzymes are two of the most important groups of biomolecules without whose active participation none of the nutrients discussed in the last three units can be utilized by the body. As a learner of dietetics, it is therefore essential for you to get the knowledge of different structural and functional aspects of enzymes and coenzymes. This unit focuses on these aspects which will enable you to understand the metabolism of different nutrients which are to be discussed in subsequent units of this course. Remember, this unit along with the next three units forms the basis component of the course. Objectives. After studying this unit, you will be able to classify enzymes and coenzymes, discuss their chemical nature and functions, describe the factors on which enzymes activity depends, and explain the diagnostic use of enzymes. So 4.2 Introduction to enzymes and coenzymes. Let us start by understanding what enzymes are. Enzymes are the proteins that catalyze biochemical reactions. Study of these important biochemical reactions was started many years ago, from the time of Louis Pasteur, who for the first time demonstrated the fermentation of glucose by yeast. The catalytic agent of yeast cell was subsequently identified and named as ferment. At the end of the 19th century, a cell-free extract of yeast was found to be capable of fermenting glucose by Buchner brothers. Considerable advances were made since then in order to properly know the nature of these agents named as enzymes. And finally, their true nature was revealed by James Summer in 1926 after the extraction and crystallization of the enzyme urease from jack beans. At present, no less than 150 enzymes have been prepared in crystalline form. What is the chemical nature of enzymes? Let's find out. We all know that enzymes are proteins. Do you recall the structure of proteins described in Unit 2 earlier? Enzymes are high molecular weight compounds made up principally of chains of amino acids linked together by peptide bond. An enzyme can be a large protein made up of several hundred amino acids or several polypeptides that act together as a unit. Enzymes have molecular weights ranging from 10,000 to 20 lakh. Until recently, it was understood that all enzymes are proteins. However, observations made in organelles from plants, yeast, viruses, and higher eukaryotic cells show that RNA can act as an enzyme. Such RNA molecules is called ribozyme. Hence, ribozymes are RNA molecules with catalytic activity. These generally involve esterification reactions and most are concerned with RNA metabolism. Recently, a ribosomal RNA component was noted to hydrolyze an amino acyl ester and thus to play a central role in peptide bond function, that is having peptidyl transferase activity. Before we move on to understanding more about enzymes, we should familiarize with the vocabulary and definitions of various terms which you will come across while studying about enzymes. Definitions of terms related to enzymes. Cofactor. Organic molecules or ions that assist many enzymes in their reactions. Apoenzymes. The protein portions of an enzyme requiring a cofactor for its reaction. Holoenzyme. A whole enzyme is a complete and functional molecule. Generally, a holoenzyme consists of a polypeptide portion and at least one cofactor or another coenzyme. Enzyme. Proteins that act as catalysts, speeding the rate at which biochemical reactions proceed but not altering the direction or nature of the reactions. Coenzymes, an organic non-protein molecule that binds with an apoenzyme, a protein molecule, to form an active enzyme. Coenzymes are often derived from vitamins. Endoenzyme, an enzyme which is not secreted or exported out of the cell but is kept and used by the cell which made it. Substrate, the specific molecule an enzyme acts upon. Metabolism, all of the organized chemical reactions in a cell which are under the control of enzymes. Catabolic reactions, reactions in which chemical compounds are broken down. Anabolic reactions, reactions in which chemical compounds are synthesized. Enzyme immobilization, the attachment of an enzyme to a solid matrix so that it cannot escape but can still act on its substrate. Enzyme inactivation, the disappearance of activity of an enzyme due to the presence of inhibitor molecules or inhibitory conditions. Enzyme kinetics, quantitative characteristics of enzymatic reactions. Enzyme stabilization, reducing the chances that an enzyme will inactive by changing the environmental conditions such as pH, temperature, concentration of salts, etc., or by attaching organic groups to it or changing some of its amino acid subunits. Having learned about the various terminologies related to enzymes, let us now continue with our study of enzymes. Enzymes are synthesized within the cell and sometimes can pass through cell membranes under certain conditions. Enzymes which act inside the cell are called as endoenzymes, and those which are liberated outside the cell are called exoenzymes. While enzymes like those involved in the production of energy belong to the first category, digestive enzymes belong to the second category. Enzymes, though basically are proteins, may sometimes have a non-protein component attached to the protein part. This may be an organic compound or a metal ion. While the first is called as coenzyme, the second is best known as cofactor. The protein part and non-protein part together form a holoenzyme, as illustrated in the figure. Sometimes the non-protein part remains so tightly bound to the protein part that it cannot be dissociated. Such non-protein part is called as prosthetic group. When the non-prosthetic component dissociates from the protein part, the enzyme loses its catalytic function and is called an apoenzyme, as can be seen in the figure 4.2. Apoenzyme plus cofactor, that is a non-protein portion, is equal to holoenzyme, means whole enzyme. Thus, coenzymes, which are usually vitamins, sometimes form an integral part of the active enzyme and make them indispensable for the enzyme to carry out the catalysis. You have already learnt about the classification of vitamins in the last unit, and you know that these are broadly classified as water-soluble and fat-soluble. Remember that water-soluble or not fat-soluble vitamins constitute the precursors of coenzymes. While participating in the mechanism of catalysis, a coenzyme undergoes alteration and its restoration requires the involvement of another enzyme. As coenzymes may participate in a variety of reactions catalyzed by different enzymes, it is also convenient to classify them on the basis of nature of element or groups such as hydrogen, carboxyl, amino, etc. They transfer in these reactions. We shall learn more about the classification of enzymes in the next section. Now let's see nomenclature and classification of enzymes. Enzymes are generally named by the addition of "ase" to the root, indicating the substrate on the which the enzyme acts. For instance, the enzymes which catalyzes the conversion of maltose into glucose is known as maltase. Many common names such as aldolase also persist, which do not tell about the substrate, although the type of reaction can be recognized. Besides, trivial names of many enzymes are in use which do not follow the convention at all, that is pepsin. In order to follow a systematic nomenclature for all the enzymes and to classify them, the International Union of Biochemistry, that is IUB, has established a system whereby the enzymes are placed into one of the 6 major classes as summarized in the table 4.1. Each class is then subdivided into several classes which are further subdivided. The system for classification of enzymes also serves as a basis for assigning code numbers to them. The code numbers are prefixed by EC, enzyme commission, and contain four numbers separated by points with the following meaning. The first number shows to which of the six main classes an enzyme belongs. The second figure indicates the subclass. The third figure gives the subclass, and the fourth figure is the serial number of the enzyme in its subclass. So you can see that a number is assigned to each class, subclass and sub-subclass so that an enzyme gets a 4-digit number. The 4-digit in fact identifies a specific enzyme. For example, alcohol, NAD, oxidoreductase is assigned as the number 1111 because it is an oxidoreductase. The electron donor is an alcohol and the acceptor is the coenzyme NAD. Thus, in naming an enzyme, the substrate is stated first following by the reaction type. The trivial name of the enzyme is alcohol dehydrogenase. Similarly, the EC number of the catalase is EC11116. The first digit 1 indicates the enzyme belongs to the oxidoreductase class 1. EC 1.1 indicates subsequent digits representing subclass and subclass. Major classes of enzymes and the types of reactions catalyzed by them are enzyme class oxidoreductase EC1. General reaction catalyzed by the class is oxidation and reduction of the substrate. Specific reactions catalyzed by the number of the class determining the subclass is first, dehydrogenases catalyze removal of two atoms of hydrogen. Second, oxidases catalyze reduction of molecular oxygen. Third, oxygenases catalyze incorporation of molecular oxygen into the substrate. Fourth, oxidative deaminases catalyze the oxidation of amino compounds with the formation of ammonia. 5. Hydroxylases catalyze the introduction of hydroxyl radical into the substrate. 6. Peroxidases act on hydrogen peroxide. Second is transferases EC2, transfer of groups between two substrates. First, aminotransferases catalyze exchange of amino and keto groups between amino and keto acids. Second, kinases catalyze the transfer of phosphate radical. Third, acyl transferases catalyzes the transfer of acyl or acetyl groups to an acceptor. Fourth, glycosyl transferases catalyze the transfer of glycosyl groups. Hydrolysis EC3 hydrolyzes the substrate. First, peptidases catalyze hydrolysis of peptide bonds. Second, glycosidases catalyze hydrolysis of glycosidic bonds. Third, esterases catalyze hydrolysis of carboxylic acid esters. Fourth, phosphatases catalyze hydrolysis of phosphoric acid esters. 5. Phosphodiesterase catalyze hydrolysis of phosphodiester bonds. 6. Deaminases catalyze hydrolysis of amines. 7th, deamidases catalyze hydrolysis of amides. Lysis EC4 removal of groups from substrates non-hydrolytically. First, decarboxylases catalyze removal of carboxyl group from the substrate. Second, aldolases catalyze removal of carbonyl group from the substrate. Isomerases EC5 isomerization of the substrate. First, racemases catalyze the conversion of D isomer to L isomer and vice versa of a compound. Second, epimerases catalyze the formation of an epimer of the substrate. Third, cis-trans isomerases catalyzes the interconversion of the cis and trans isomer of the substrate. Fourth, aldose-ketose isomerases catalyze the conversion of aldose and to ketose and vice versa. 5. Mutases catalyze the intermolecular transfer of the group. Ligases EC6 joining of two substrates at the expense of the breakdown of one pyrophosphate bond of ATP. First, synthetases catalyzes the formation of carbon-oxygen bond, carbon-sulfur, carbon-nitrogen bonds at an expense of ATP. Second, carboxylases catalyzes the introduction of carboxyl group with a carbon-carbon bond formation at the expense of ATP. From the classification above, the different classes of enzymes identified include oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Let's get to know more about them and their nomenclature. Oxidoreductases. To this group, EC1 belongs all enzymes catalyzing oxidation reduction reactions. Common names includes dehydrogenases, oxidases, reductases, and catalysases. The substrate AH2 that is oxidized is regarded as an hydrogen donor. AH2 plus B is equal to A plus BH2. The recommended name is dehydrogenase, but as an alternative, the term reductase is used. The name oxidase is restricted to enzymes which exclusively use O2 as an hydrogen acceptor. The second figure in the code number of oxidoreductases indicates the group in the hydrogen donor which undergoes oxidation. For example, CHOH, CHO, CH bond, CHNH2, NADPH. Therefore, EC1.1 indicated acting on the CHOH group of donors. EC 1.2 acting on the aldehyde or oxo group of donor, etc. The third figure indicates the type of acceptor involved. 1 denotes NADP, 2 a cytochrome, 3 O2, 4 a disulfide, 5 a quinine, etc. So EC 1.1.1 states with NAD positive or NADP positive as acceptor. EC 1.1.2 with cytochrome as acceptor, and so on, the list goes on. Transferases. These are the enzymes code EC2 which catalyze the transfer of a group, for example a methyl or glycosyl group, from one compound to another. In many cases, the donor is a cofactor, coenzyme carrying a group to be transferred. Common names include acetyltransferase, methylase, protein kinase, and polymerase. The second figure EC 2.1 in the code number of transferases indicates the group transferred. A carbon group 2.1, a carbonyl group aldehydic or ketone 2.2, a glycosyl group 2.3, and so on. The third figure informs of the group transferred. For example, subclass 2.1 is subdivided into methyl transferases 2.1.1, hydroxyl methyl and formyl transferases 2.1.2, and so on. Now, hydrolysases. These enzymes coded EC3 catalyzes the hydrolytic cleavage of carbon-oxygen bond, carbon-nitrogen bond, carbon-carbon bond, and some other bonds, including phosphoric anhydride bonds. Their trivial names are formed by adding the suffix ase to the substrate which they hydrolyze. Examples include protease, nuclease, phosphatase. A number of hydrolyses acting on ester, glycosyl, peptide, amide, or other bonds are known to catalyze not only to hydrolytic removal of a particular group from their substrate, but also the transfer of this group to a suitable acceptor molecule. Yet they are not grouped as transferases because the transfer of a specific group to water as the acceptor molecule is considered to be their main physiological function. The second figure in the code number of hydrolysis indicate the nature of the bond hydrolyzed, that is, esterases, 3.1, glycosidases 3.2, and so on. The third figure generally specifies the nature of the substrate, that is carboxylic ester 3.1.1, thiol ester 3.1.2, phosphoric monoester 3.1.3, O glycosides 3.2.1, N glycosides 3.2.2, and so on. Lyases. These enzymes code EC4 cleave carbon-carbon bond, carbon-oxygen bond, carbon-nitrogen bond, and other bonds by elimination, forming double bond or conversely adding groups to double bonds. Common names include decarboxylase, aldolase, dehydratase if water is eliminated, or hydrolyase if the reverse reaction is more important or the only one which can be demonstrated. Synthase but not synthetase may be used as in tryptophan synthase or cystathionine beta synthase. The second figure in the EC number indicates the bond being cleaved, for example, carbon-carbon lyase 4.1, carbon-oxygen lyase 4.2, and so on. The third figure informs about the group that is eliminated, for example, CO2 4.1.1 or H2O 4.2.1. Isomerases. These enzymes code EC5 catalyze geometric or structural change within a molecule according to the type of isomerism. They may be called racemases, epimerases, cis-trans isomerases, isomerases, tautomerases, mutases, or cycloisomerases. EC 5.1 are racemases or epimerases. EC 5.1.1 indicates acting on amino acids and derivatives and so on. Ligases. These enzymes code EC6 catalyzes the linkage of two molecules coupled with the hydrolytic breakdown of a pyrophosphate bond in ATP or an analogous compound. The bonds formed are often high energy bonds. The second figure in the code number indicates the bond formed, that is carbon oxygen 6.1, carbon sulfur 6.2, etc. Examples include peptide synthase, amino acyl tRNA synthase, DNA ligase, and RNA ligase. We hope the information given above may have helped you in understanding the classification of enzymes. The system of the nomenclature and the classification of enzymes are based exclusively on the reactions that is catalyzed and does not consider their origin or multiplicity. Enzymes catalyzing the same reaction but isolated from different species will have varying amino acid sequences so that they may be distinguished by electrophoretic methods. They may have different sizes and net negative charges and may even differ in their catalytic behaviors. Specificity of enzymes. One of the major characteristics of enzymes is that they are highly specific. Enzymes, the organic catalysts, differ from the inorganic catalysts in their extraordinary specificity. Each of the enzymes that have been isolated and studied is found to possess different types of specificities. A few enzymes exhibit absolute specificity, that is, they will catalyze only one particular reaction. Other enzymes will be specific for a particular type of chemical bond or functional group. In general, there are four different types of specificity. These are as follows: reaction specificity, bond specificity, group specificity, and optical specificity. We shall look at each of these specifications one by one. First, reaction specificity. Some enzymes catalyze only one reaction acting on a specific substrate. For example, urease and catalase acts only on urea and hydrogen peroxide respectively. This is called absolute specificity. Second, many enzymes can catalyze same type of reactions: phosphate transfer, oxidation reduction, hydrolysis, etc. in several structurally related compounds. Example, carboxypeptidase acts on protein chains and removes one amino acids at a time from the C-terminal, irrespective of the nature of amino acids. Third, a substrate can undergo many reactions, but in specific reaction an enzyme will catalyze only one of these reactions. Example, citrate synthase convert oxalatoacetate to citrate in the presence of acetyl coenzyme A, but in absence of acetyl coenzyme A, oxaloacetate is acted upon by the different enzyme maltase dehydrogenase with the formation of malate. Bond specificity. First, some enzymes acts on a particular bond, glycosidid, peptide, ester, etc. Examples: pepsin, trypsin, chymotrypsin, etc. are the only acting on peptide bond. Through the peptide bonds on which they will attack depend upon the amino acids that have formed the bonds. Group specificity. Some enzymes prefer a specific functional group to be present on the substrate molecules. Example, alcohol dehydrogenase acts on alcohols having OH group. Second, some proteases acts on the peptide bonds depending on the amino acids involved in the formation of the bonds. Example, chymotrypsin hydrolyzes only the peptide bonds which is formed by the carboxyl group of phenylalanine, tyrosine or tryptophan. Optical specificity. Some enzymes exhibit absolute optical specificity for at least a portion of the substrate molecule. For example, the enzyme maltase catalyzes the hydrolysis of alpha glycosidic bond between two glucose molecules but not the beta glycosidic bond. Second, enzymes are able to discriminate between the optical isomers and can act accordingly. Example, L-amino oxidase acts only on L-amino acid and not on D-amino acids. Having gone through the section above, it is clear that enzymes can be very specific. Next, let us learn about the mechanism of action, but first let us recapitulate what we have studied. Mechanism of enzyme action. L. Michaelis and M.L. Manton developed a general theory of enzyme action in 1913. According to Michaelis and Manton, the enzyme first binds with substrate to form a transient enzyme substrate complex. This complex then dissociates into the product and the unaltered enzyme. Let us understand how this ES complex forms. The action of an enzyme is initiated when the reactants, that is substrates, bind at the catalytic sites or active sites on the enzyme molecules. The catalytic sites of the enzyme molecules possesses a complex three-dimensional form and provides a cleft which binds the substrate as shown herewith. A change in the tertiary or quaternary structure of the enzyme may alter the three-dimensional shape of the catalytic site and thus reducing its binding and catalytic activities. The ES complex is formed mainly by non-covalent bonds between specific groups of the substrate molecules and the specific amino acid side chains present in the catalytic site of the enzyme. Different models for enzyme substrate complex formation exist. Let us look at these models next. Models for enzyme substrate complex formation. There are two popular models to explain the enzyme substrate interaction. These are: first, Fisher's template or lock and key model. Second is Koshland's induced fit model. The two models are described next. Fisher's template or lock and key model. According to this model, the catalytic site of the enzyme has a proper conformation compatible to a specific substrate even in the absence of a substrate molecule. As shown in figure 4.3, the catalytic site binds the substrate and catalyzes the reaction without any change in its own three-dimensional conformation. It has become possible to explain the specificity of many enzymes for only one of the stereoisomers of the substrate by this model. This model, however, failed to explain the change in the enzyme activity in presence of allosteric modulators, low molecular weight regulatory substances that bind at a specific site on the enzyme molecule other than the catalytic site, and thereby enhances the or inhibit the enzyme activity or the action of the non-competitive inhibitors, about which we will learn later in the section 4.8 in this unit. Now let's see the second model, that is Koshland's induced fit model. This model considers a flexibility in the three-dimensional conformation of the catalytic site. According to this model, despite having a required amino acids, the catalytic sites of the Enzyme does not possess the conformation complementary to the substrate in absence of the substrate molecule. Only when the substrate approaches towards the enzyme or during its binding, the conformation of the catalytic site changes so that the enzyme can hold the substrate properly. This model therefore can suitably explain the non-competitive exhibition and non-competitive inhibition and allosteric modulation of the enzyme. We will learn about the non-competitive inhibition later. While on the topic of the mechanism of enzyme action, it is also important to know about the unit of enzyme activity, that is, how to express the activity of an enzyme. Have you ever heard of the term kettle? Recommended unit of enzyme activity is called as kettle, which is the amount of an enzyme that transforms 1 mole of substrate into product in 1 second. In fact, other than kettle, the activity of an enzyme may be expressed in different other ways as highlighted herewith. A. International Enzyme Unit IU is defined as the amount of enzyme that catalyzes the transformation of 1 mu mole of substrate into product in 1 minute. Specific activity of an enzyme preparation is expressed as cat per kg or IU per mg of protein. C. Molar activity of an enzyme is cat per mole of the enzyme. D. Turnover number of the enzyme is the number of the molecules of substrate transformed per catalytic side of the enzyme per minute. After understanding the mechanism of enzyme action, we move on to the enzyme kinetics. What is enzyme kinetics? Let's find out. The study of the rate at which an enzyme works is called enzyme kinetics. You have read in the last section that L. Michaelis and M.L. Menton developed a general theory of enzyme action and kinetics in 1913. According to this theory, we learned that in an enzyme-substrate reaction, the enzyme first combines with the substrate with the formation of an enzyme-substrate complex, which subsequently break down into product and the enzyme is recovered. Thus, the enzyme catalyzed reaction proceeds in the following 2 steps. The reactions are assumed to be reversible and K1, K2, K3 and K4 are the rate constants in each reaction. Let us examine enzyme kinetics as a function of the concentration of substrate available to the enzyme. The enzyme substrate interaction may be studied with the help of figure 4.5. In this graph, substrate concentration and velocity of the reaction of the enzyme catalysis has been shown on x and y axes respectively when the velocity of the reaction is plotted against different substrate concentrations as in the present graph. A hyperbolic curve is obtained. M, N and O are the 3 points on the curve representing 3 stages of enzyme catalyzed reaction, while M represents the stage of the reaction when the substrate concentration is very low and the rate of the reaction is directly proportional to the substrate concentration. O represents the stage when the reaction velocity reaches its maximum due to the gradual increase in substrate concentration resulting in the saturation of the active sites of the enzyme. In between these two extremities lies the third point N, which represents the reaction velocity of the enzyme catalyzed reaction, that is half of the maximum velocity. Now let's see factor affecting enzyme activity. Rate of an enzyme substrate reactions depends on several important factors. These includes concentration of enzyme, concentration of substrate, pH, temperature, etc. Without the optimum conditions of these factors, the enzymes will be unable to exhibit its best activity. These factors are discussed briefly in this subsection. Concentration of enzymes. Increase in enzyme concentration will increase the rate of the enzyme catalyzed reaction. This is because of the availability of additional catalytic sites to which the substrate can bind. Accordingly, a straight line graph is obtained as can be seen in the figure. Since the concentration of the substrate relative to that of the enzyme is always very high, increase in enzyme concentration results in increased enzyme activity. Concentration of substrate. When the concentration of the substrate is low, the rate of the enzyme catalyst reaction always remains low. In spite of concentration of substrate being higher compared to enzyme concentration, this is because at this stage, all the catalytic sites of the enzyme are not occupied by the substrate molecules. So with the increase in substrate concentration, the rate of the reaction also increases until all the catalytic sites of the enzymes are utilized. Rate of the reaction becomes maximum at this point, and beyond this it remains constant as can be seen in the figure. Temperature. The rise in temperature accelerates the rate of enzyme catalyzed reaction up to a certain temperature known as optimum temperature for the enzyme. At very high temperature, the enzyme undergoes denaturation and subsequent loss of activity. For most enzymes, the optimum temperature are close to that of ambient temperature of the cell. For human beings, the temperature is in the region of 37 degrees Celsius, while the optimum temperature in plant urease is 60 degrees Celsius. Certain microbial enzymes have a higher optimum temperature that enables them to adjust in a new higher ambient temperature of a new environment. The effect of temperature on the reaction rate is shown in the figure. You can see that the rate of the reaction is almost zero at 0 degrees Celsius, and this gradually increases with the rise of the temperature until the optimum point reaches. Beyond this point, the activity of the enzyme falls due to denaturation of the curve bend, reaching ultimately the zero level. It has been found that the rise of 10 degrees Celsius will double the activity of the enzyme. pH. Enzymes are influenced by pH changes as they have a ionic character due to the presence of amino and carboxylic groups. All enzymes have an optimum pH for showing highest catalytic activity, and a change of this affects their activity. However, with a narrow pH range, the change in the reaction rate are reversible, but if the pH becomes too low or high, denaturation of the enzyme may occur. Most enzymes have an optimum pH range between 5 and 8. Although some enzymes like pepsin and trypsin are most active at high acidic pH 1.5 and high alkaline pH 8 condition. A typical curve of enzyme activity against pH changes is presented in figure. Role of enzymes and coenzymes in metabolism. You have already learned that many enzymes requires a non-protein part for their optimal activity, which may be a coenzyme or a metal ion. This has also been stated earlier that many water-soluble vitamins are acting as coenzymes in their native or derived forms. So it is important to learn how the coenzymes are assisting different enzymes in catalyzing biochemical reactions. In this subsection, you will learn about the chemical nature of different coenzymes and the general role of coenzymes in assisting different enzymes during metabolism. As coenzymes participate in a variety of functions, they can be classified broadly into two types. First, hydrogen transferring coenzymes, and second, group transferring coenzymes. Let's learn about these two groups of enzymes. First, hydrogen transferring coenzymes. This group consists of three important coenzymes, all of which assist different enzymes in oxidation reduction reactions. These are: first, nicotinamide nucleotide; B. Flavin nucleotides; third, lipoic acid. A brief review of these coenzymes follows. Nicotinamide nucleotides. These coenzymes are derived from the vitamin niacin. You may recall reading in Unit 3 that they are of two types: nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. Collectively, they are called as pyridine nucleotides. Have a look on the figure. In NAD positive, the pyridine ring is attached to the ribose molecule through glycosidic bond, and the phosphate provides a link between adenosine and nicotinamide riboside. In NADP positive, an additional phosphate group is present in carbon atom 2 of the ribose molecule of adenosine component. Both NAD positive and NADP positive act as hydrogen acceptors during oxidation reduction reactions in the body. Dehydrogenation is the primary mechanism of biological oxidation in which two hydrogen atoms are removed from the substrate in presence of an acceptor. The hydrogen atom ionizes to yield 2H positive and 2 electrons. The nicotinamide ring of NAD positive or NADP positive accepts a proton and 2 electrons, which are equivalent to H negative. The other H positive remains as such. All reactions catalyzed by them are reversible. Table 4.3 lists some of the enzymes, all dehydrogenases, which are dependent on these coenzymes. Firstly, we will say enzyme, then coenzyme, then substrate, and then product. First enzyme is lactate dehydrogenase, NAD positive, lactate pyruvate. Second is alcohol dehydrogenase, NAD positive, primary and secondary alcohols, corresponding aldehydes and ketones. Glutamate dehydrogenase, NAD positive, glutamate, alpha ketoglutarate, and ammonia. Glyceraldehyde 3-phosphate dehydrogenase, NAD positive, glyceraldehyde 3-phosphate, 1,3-diphosphoglyceric acid. Glucose 6-phosphate dehydrogenase, NADP positive, glucose 6-phosphate, 6-phosphogluconoloctone. Malate dehydrogenase, NAD positive, malate, oxaloacetate. Isocitrate dehydrogenase, NADP positive, isocitrate, alpha ketoglutarate. Second is flavin nucleotides. Flavin nucleotides are derived from the vitamin B2, riboflavin, and are actively involved in hydrogen transfer reactions. You may recall reading in the last unit about the two coenzymes and the product produced from riboflavin, that is flavin mononucleotide FMN and flavin adenine dinucleotide FAD. Look at the figure 4.16. For the structure of FAD, FMN is the active component of riboflavin and is formed by the addition of a phosphate group, and FAD is formed by the combination of FMN and with one molecule of adenosine triphosphate. Both FMN and FAD are involved in many oxidation reduction reactions. Enzymes that require the presence of FMN and for their catalytic activity include glycolic acid oxidase, L-amino oxidase, etc. FAD containing enzymes are succinate dehydrogenase, D-amino oxidase, etc. Third is lipoic acid. Lipoic acid acts in the transfer of hydrogen during oxidative decarboxylation reactions. It occurs both in oxidized and reduced forms. For its structure, see in the figure. This figure shows the structure of lipoamide where lipoic acid is bound in an amide linkage to the E-amino group of the lysine residue, that is blue, of dehydrolipoamide acyl transferase. The complex reactions of the carbohydrate metabolism catalyzed by pyruvate dehydrogenase system and the alpha ketoglutarate dehydrogenase system requires the participation of lipoic acid. These reactions will be discussed later in the unit covering carbohydrate metabolism. Second, group transferring enzymes. This group consists of the following coenzymes involved in different metabolic reactions where the transfer of any functional group is taking place. Biotin, thymine diphosphate, pyridoxal phosphate, coenzyme A, THF, cobamide coenzymes, ATP. You may recall reading about these enzymes in the last unit on vitamins, particularly under the section on water-soluble vitamins. There we talked about them as derivatives of water-soluble vitamins. We shall look at these enzymes once again to help you understand them better. First, biotin. This water-soluble B group vitamin participates in the transfer of carboxylic acid. Two important enzymes carrying out carboxylation reactions are pyruvate carboxylase and acetyl coenzyme A carboxylase. Pyruvate carboxylase contains 4 biotin molecules attached to the enzyme protein. The biotin dependent enzymes require ATP, which is converted into ADP during the reaction. Then TDP, thiamine diphosphate. Thiamine diphosphate is the coenzyme responsible for the transfer of aldehyde and glyoxal groups. The most important metabolic reactions catalyzed by TDP is the oxidative decarboxylation of alpha keto acids such as pyruvic acid. The involvement of TDP in the decarboxylation of pyruvic acid was so well known that the coenzyme was also popularly called as co-carboxylase. It is also actively taking part in the conversion of pyruvic acid to acetyl coenzyme A and alpha ketoglutarate to succinyl coenzyme A that are catalyzed by pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase complex respectively during carbohydrate metabolism. You shall learn about this in the more unit. Another important metabolic reaction catalyzed by TDP is the conversion of xylulose 5-phosphate to pseudoheptulose 7-phosphate catalyzed by the enzyme transketolase in pentose phosphate pathway of carbohydrate metabolism. Look up in Unit 6 for knowing more about the pentose phosphate pathway. Next is pyridoxal phosphate. Pyridoxal phosphate is derived from the pyridoxine vitamin B6 and is involved in amino acid metabolism. The other two compounds, pyridoxal and pyridoxamine, about which you learned in the last unit, having the properties of vitamin B6, also occur as phosphate derivatives. Enzymes that are dependent on B6 phosphate coenzyme catalyzes a variety of reactions such as transamination, transfer of amino group from an amino acid to a keto acid, decarboxylation, removal of carboxyl group, and racemization, transfer of one isomer to another. The structure of this is shown in the figure below. Enzyme glutamate oxaloacetate transaminase catalyzes the reaction between glutamate and oxaloacetate with the formation of alpha ketoglutarate and aspartate due to the transfer of one amino group from glutamate to oxaloacetate. Pyridoxal phosphate acts as the carrier of the amino group whenever it accepts the amino group. It transforms the pyridoxamine phosphate and after the release of the amino group it again becomes pyridoxal phosphate. Aspartate is decarboxylated by aspartate decarboxylase taking the help of pyridoxal phosphate. L-alanine transforms to D-alanine by alanine racemase, which also requires pyridoxal phosphate as the coenzyme. Coenzyme A. Coenzyme A is derived from the vitamin pentathonic acid. This is abbreviated as CoA. This can be divided into two components, adenosine 3,5-diphosphate and pentothene, which is formed by the combination of pantothenic acid and mercaptoethyl amine. Refer in the figure to understand its structure well. It gives rise to acetyl coenzyme A, derivatives that are mainly formed in ATP-dependent synthetase reactions. These are highly reactive and participate in various types of reactions. For example, oxaloacetate is converted to citrate in presence of citrate synthase by accepting acyl group of acyl coenzyme A during carbohydrate metabolism. The pyruvate is converted by the pyruvate dehydrogenase complex to acetyl coenzyme A by the active participation of coenzyme A. Also, in the oxidation of fatty acids, coenzyme A has an important role to play. You will find out in the next unit. Adenosine triphosphate. Though ATP often functions as a second substrate, it can also serve as a coenzyme by modulating the activity of specific enzymes. The compound consists of adenine connected to a ribosyl 5 triphosphate. As a co-substrate, ATP is utilized by the various kinases for the transfer of the terminal phosphate group to various acceptors. For example, glucose is converted to glucose 6-phosphate in presence of the enzyme hexokinase by accepting phosphate from ATP. This will be discussed many times in Unit 6 and 7 covering carbohydrate and lipid metabolisms. Thank you.